Hey y'all, Brown's back, and we're going to learn some more about the histology of the alimentary canal, this time focusing on the small intestine. So your small intestine is kind of the transition between some important digestive processes. Digestion, both chemical and physical digestion, is completed in the small intestine. The small intestine is also where the vast majority of absorption takes place. So this is going to be the spot where all of those nutrients that your mouth and your stomach and your small intestine have broken down into their simplest forms, monosaccharides, nucleic acids, amino acids, etc., are going to transition from being in what is the external environment of the lumen of the alimentary canal to the internal environment. We're going to get this stuff into the bloodstream or the lymphatic system so that our bodies can actually use it. So the modifications that we're going to see in the small intestine are largely geared toward that absorptive property. You'll see what I mean in just a second. So if we look at the wall of the small intestine, it is highly folded. So the mucosa is folded into tiny little finger-like projections called a villus, and then in part of the small intestine, the middle part, the mucosa and the submucosa are both folded into large structures called plicae circularis. These are going to make the intestine have an almost corrugated appearance. It's going to look ribbed or rippled. Think about those like black plastic drain pipes that you often see sometimes. That's kind of what the inside of your intestines look like. But let's focus a little bit more on the villus here. So a villus is a finger-like projection, so you can imagine it comes up like a O. So we're going to zoom in on that single villus. And then we'll go up like this. And this is going to be the epithelium. So I told you these villi being the plural of villus, are modifications of the mucosa. So we have our epithelial cells here, and that one bled over a little bit. Y'all don't understand how hard it is to draw on this tablet while you're working on a computer. Anywho, so these are the epithelial cells, poorly drawn, but there they are. And you can see that by folding this up, we're packing a lot of epithelial cells into a rather small space. The small intestinal mucosa is modified throughout to increase the surface area of the mucosa exposed to the lumen, thereby increasing the amount of absorption we can occur. Now, inside this villus, in the lamina propria here, right, because this is the mucosa, so we have an epithelium with a lamina propria underneath it. In the lamina propria down here, we're going to find two blood vessels. So there's going to be a little artery, and that's going to supply blood to the cells of the mucosa. We're going to see a little vein. This vein is going to transport away all of the proteins, carbohydrates, and nucleic acids that were absorbed by the mucosal cells. So stuff is going to go from out here through this mucous membrane and into here, where it then gets absorbed into this vein and is transported to the liver for processing. Finally, there's also a lymphatic vessel in here called a lacteal. So we'll put him maybe right in here. Absorbed fats are actually going to go into the lacteal, the lymphatic vessel, inside the villus. Now this looks like a lot of folding, doesn't it? We've got the epithelium folded into these villi, and then we can fold the whole mucosa up into these plicae circularis. But there's actually more folding going on. So if we look at a micrograph of a small section of the small intestine, we see that it is a simple columnar epithelium. It is a goblet cell epithelium, so here we can see a goblet cell. But what I really want to draw your attention to is the apical surface of the mucosal epithelial cells, right here. See how it looks kind of fuzzy? While these cells are in a villus, right? So imagine that thing I drew in the last picture but turned sideways. So here's the lamina propria down here. Here are the mucosal cells here. So just outline a single cell. This little guy's doing like this. This fuzzy bit up here at the top, these are structures called microvilli. And the microvilli are actually folds of the apical plasma membrane. So not only are we folding up the mucosa and folding up the epithelium, we are folding up the membrane on the mucosal epithelial cells. So let's look at this even more closer. Let's bring out the old TEM. Oops, erase the rest of this too so we can see it better. Y'all, I'm trying to make this online experience as much like in-ground as possible, complete with derps on the board. So now we can see this is a single epithelial cell, and you can see that the plasma membrane is highly folded. Folds on top of folds on top of folds on top of folds. All of this, good at even higher magnification, is going to vastly increase the surface area. Let's think about what we've done here. Imagine this is just a sheet of epithelial cell membrane. Well, by folding that membrane up into microvilli, we're going to get something like this. So we're going to get the same amount of membrane, but in a much smaller space. And then we're going to fold this up into a villus. So now we've gone from taking up the whole width of the screen to this much of the screen to this much of the screen, and then we take those villi and pack those into plicae. This would be a much zoomed out version. What you can see now is that we can fit, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 of these into a fairly small space. So all of this folding is going to vastly increase the surface area of the mucosa exposed to the lumen, and since to be absorbed something has to cross a cell membrane, the more cell membrane we can expose per centimeter of intestine, the more efficient absorption is going to be. So most of the modifications of the small intestine that happen throughout the small intestine are happening at the mucosal layer. Once we get into the 3 regions of the small intestine, I'll show you some modifications that happen in the submucosa. Muscularis is pretty regular the whole way out, but serosa is also modified. If we look here, we can see in this picture of, I think this is a cat intestine, it's been colored to highlight the structure called the mesentery. And the mesentery is kind of an outpinching of the serosa. So if we look at the light micrograph on the right, you can see the mucosa, farthest on the left, submucosa, muscularis externa, and here is the serosa. So here's the inside of the serosa. I'll mark off the border between muscularis externa and serosa. But see how the serosa gets pinched out here? This outpinch structure is the mesentery. This is this membrane that we're seeing over here. And it is from the mesentery that your intestines are suspended. They're not just hanging out all loose in there. They're actually suspended from the peritoneal wall by this outpinching of the serosa. An analogy I often use is, imagine like in an old Robin Hood movie where somebody would get an arrow shot through their shirt and it would kind of pin them to a tree. That's kind of how your intestines are suspended from the peritoneal wall. The last thing I want to do to try to keep this video under 10 minutes is go through the 3 sections of the small intestine, because it is divided into 3 distinct histological sections, and show you the defining feature of each section and talk a little bit about why each section needs that defining feature. So the most proximal portion of the small intestine, this is the one closest to the stomach, is called the duodenum or duodenum, depending on if you prefer the the British pronunciation duodenum or the more American duodenum. Either way is fine with me. I'm probably not going to hear you say them. The duodenum, you can see in this micrograph, has villi. So here are our villi poking up. It's down in the submucosa, however, that we see kind of the defining feature, the thing that lets you identify this as duodenum and not some other part of the small intestine. And that is these coiled tubular glands visible here at the end of the microscope's pointer. And these are called Brunner's glands. And Brunner's glands, they're kind of like the Skrillex of the small intestine. If you don't remember who Skrillex is, he is a dubstep, like EDM, electronic dance music producer. And what happens in the middle, or 40 seconds into every EDM song? They drop the bass, and that's what these guys do. Remember, the duodenum is right next to the right next to the stomach. So the chyme, that paste that was formed in the stomach that's coming into the duodenum, is going to be highly acidic. And these Brunner's glands are going to secrete bicarbonate. And bicarbonate is a base. It's going to scavenge those free protons to produce carbonic acid, which is much less acidic. So we're getting rid of the acid. So duodenum, submucosa, is going to have Brunner's glands. You're not going to see these anywhere else in the small intestine. If we look in the submucosa of the jejunum, the middle portion, we're not going to find a whole lot. You just got a whole lot of nothing down in there. Switch pen colors to something that's a little easier to see here. So you got nothing in the submucosa. You have some connective tissue. You're going to have arteries and veins, but there are no real specializations in the submucosa of the jejunum. By the time the food products, the chyme, makes it into the jejunum, digestion is complete. All the acids have been neutralized, and it's the job of the jejunum to do absorption. This is the most important absorptive part of the small intestine. You can see the prominent villi up here at the top. You may also see some white spots in there. Those are goblet cells. We're going to see a lot more of those, especially when we get into the large bowel. So the jejunum is highly absorptive, so it doesn't have a whole lot going on in the submucosa. You can see that over here as well. There's the whole submucosa. We have some connective tissue, got a little arteriole going on here, not a whole lot else. However, as we make our way further along and we enter the third, the most distal portion of the small intestine, we make our way into the ileum. The ileum is the part that connects to the large intestine. And in case you didn't remember, the large intestine contains a ton of bacteria. In fact, you have more bacterial cells in the lumen of your large intestine than you have human cells and all your tissues and organs added together. And although there is a valve that prevents stuff from bleeding back from the large bowel into the small bowel, into the ileum, there can still be some transfer of bacteria from the large intestine to the small intestine, and if that happens, it's going to happen to the ileum. This is the part where it could be exposed to bacteria. In the submucosa of the ileum, we see these structures circled in red in the micrograph on the right, and these are called Peyer's patches. Peyer's patches are lymphatic tissue. These are full of macrophages and lymphocytes, immune cells, that can take care of any bacteria that make it across this epithelium. Remember, out here, this is external. Your immune system doesn't care about what's going on out here. But as soon as something makes it across this epithelium and into here, it is now internal and therefore could cause infection. So your ileum of your large intestine is going to contain numerous Peyer's patches. So here's another micrograph, and you can see the very large, distinct Peyer's patches present in the submucosa. So just to recap, the small intestine completes digestion and most of absorption. Because it is absorptive, it has all of these modifications to increase the surface area of mucosal cell membrane presented to the lumen, starting with microvilli, those foldings of the plasma membrane, going to villi, which you can see here, and then ending with the plicae circularis predominant in the jejunum. Three regions of the small intestine: the duodenum with its Brunner's glands in the submucosa, the jejunum with a submucosa containing primarily vasculature because it's doing the absorption, and then the ileum with the Peyer's patches in the submucosa. Sorry this video went over 10 minutes, but I had a lot to tell you. I hope you enjoyed it. Have fun, study hard. We'll see you later.